Okay, so I'm just going to explain about a current project I'm working on now, which is on understanding how cells or groups of cells express themselves genetically. So, for instance, in our human body, we have lots of different cells that are doing lots of different things. We have our gut cells, our skin cells, probably some stuff, other cells. There's lots of cells all around. But all of these cells have genes that come from the human genome. So the human genome is like the collection of all the genes that humans have. And what are genes? Genes are a little piece of DNA that roughly translates to a protein. And it's by producing proteins that our cells do their everyday thing. Either the immune system or hair. Are those proteins? I don't know. They're kind of like proteins, but mostly, that's what cells do. They make proteins and the proteins do their stuff. Okay, so what biologists and scientists want to be able to do is they want to get like a little sample of cells, maybe from the sewers to see how people's gut testing is, or maybe from your lungs to see how they're doing, or from any population, just a group of cells from say humans, and you want to know what genes are being expressed by those cells. And if you knew what genes are being expressed, you know what proteins those cells are making, and you can have an idea of what they do. How do they work? Okay, so that's the gene expression problem. Given some cells, find out which genes of the human genome are they activating, are they using? So this is not so easy, because when you get a bunch of cells and you collect their genetic code, you can get the genetic code from a bunch of cells and you can amplify it in a sample. Well, amplify it just means that you sort of like make whatever genetic code was in those cells replicate many times. So you have like 1,000 times more genetic code than when you started. And the way that we then measure what genes are in the genetic code is we put them into this fancy RNA sequencing machine that chops up the DNA into little pieces. It's like a crazy Japanese chef with knives chopping it up into little pieces. And it chops it up into pieces in a way that doesn't respect, what is a gene and what's not a gene. It just chops it up into pieces and doesn't care that, this is a gene. It's just gonna be, I don't care. Hiya! Cuts it in half. So you end up with lots of these fragments of genes after you put your sample into this fancy RNA sequencing machine. And the puzzle then is, given all these chopped up little pieces of genes and DNA, how can you figure out what genes were originally in your cell population? Now, this turns out to be a fundamentally challenging numerical problem. In fact, this is the kind of problem that we call a combinatorial problem, because there are a huge number of ways in which you can chop up DNA. There are exponentially many different ways in which you could have cut up that DNA into pieces. So when you want to reconstruct what was that original DNA, there are like so many ways you could stick together those little chopped up pieces again to form genes. So this has been a real challenge to be able to just numerically scale up and solve and figure out what genes are being expressed because of this. So the project I'm working on, which I'm not leading at all, I am just an extra, I'm just a collaborator, is we're trying to use some advanced statistical tools. And we figured out that we can describe mathematically what are the most likely ways that genes and genetic code will be chopped up by things like the RNA sequencing machine. And once we have a mathematical model of what's the most likely way these genes have been chopped up, we can then look at the little pieces of chopped up DNAs, genetics, and figure out what was the most likely genes that produced those little pieces. So we actually solve an optimization problem that says, give me what were the most likely genes that produced these tiny chopped up little pieces of DNA. And that's where I come in, because I'm an optimizer. So I'm figuring out mathematically what is possible, answering questions, can we solve this optimization problem? And the answer is yes, we can. Then I'm writing code to do this at scale, because the human genome is massive. It's like I'm going to say a number, I think 800,000 genes, but I'm not totally sure. It's a lot. Okay, it's a lot. So then I'm writing code and trying to give everything, the mathematics, the code, and everything to solve this problem that will tell you what are the most likely genes that are being expressed.